Hey guys, welcome back. Today I am in the classroom and I apologize if my audio isn't fantastic. I forgot my microphone and it's very echoey in here. Alas, we must make do with what we have. And so today I'm going to talk about transport again, right, cell transport, and I'm going to be looking at more specifically the mechanisms of passive transport and the mechanisms of active transport. So last time was a bit of an introduction, and this time we're going to look a little bit more in detail at how these things work. So passive transport as a general concept is when we are moving molecules in or out of the cell without using any energy. And it's driven entirely by the principles of diffusion. So these molecules are always moving from an area of high concentration to an area of low concentration. Yeah, that's what's happening here. And there are two types of passive transport. Simple diffusion occurs when the molecule that we're moving is small and nonpolar. So these small nonpolar molecules can just go straight across the membrane without any issues. Okay? They're smaller than the gaps between the phospholipids that make up the membrane. And because they are non-polar, they get along with the tails of these phospholipids, and so they don't reject them. Right? So these small non-polar molecules just slip right through the gaps. Okay? So the example molecule I've used is oxygen gas, the oxygen that we breathe in. It's non-polar because it's a very balanced, even molecule. Right? Both ends are exactly the same, so there's no charge on either side. Okay? They're well-balanced molecules, they're tiny, so they just go right through this gap right here. Well, so in this model, I have a high concentration of oxygen outside of my cell, in what's called the extracellular region, extra meaning outside of, okay? And a lower concentration inside my cell, or the intracellular region, right, inside the cell. Okay? And so these oxygen molecules, as a net, are going to move into the cell. Right? They're just going to pass right through until I have roughly equal amounts inside and outside. We use a state we call equilibrium balanced. Okay? So that's simple diffusion. Small nonpolar molecules go right across the membrane from high concentration to low concentration. Cool? Still in passive transport, we're now going to look at what's called facilitated, which means to help, right? To facilitate means to help. Diffusion. Now these are molecules that are larger, molecules that are polar, or molecules that are ionic, which is charged molecules, if it's a plus or a minus molecule. Right? So like if you have table salt, the delicious stuff we put on our food, that's NaCl. And it's ionic because the sodium, Na, is Na+, and the chloride, Cl, is negative. And so I have a cation and an anion, plus charge and negative charge. Those things also cannot just go straight across the membrane. Okay? So if I have a large molecule, a polar molecule, or an ionic molecule, they need help getting across the membrane. They can't go through the gaps. And what's going to help them make it across the membrane are going to be protein channels. So in the cell membrane, there are embedded these proteins, and they have a tube hole in them so molecules can go through. Okay? They pass across the entire membrane, they connect the outside and the inside. These protein channels have specific shapes, right? And so these shapes match certain molecule types. And in my model here, I have a protein channel that's allowing these sugar molecules to go through. Sugar's big, it's too big to fit in the gap, so it has to go through this protein channel. Okay? And so I have a high concentration up here, a low concentration down here. The sugar molecules move through the channel to go inside the cell. These are waters, right? These H2Os. These are tiny. They're small enough to go through the gaps, right? But water is polar. Right? It has a slight positive charge on the hydrogen side and a slight negative charge on the oxygen side, principle we talked about at length in the beginning of the year, right? Because the oxygen is electronegative, the electrons hang out near the top more often than near the bottom. So this side's kind of negative, this side's kind of positive. The lipid part of the phospholipids don't like that. Right? So if this water tries to go in through the gaps, right, when it hits the lipid part, the lipid says, get out of here, and it just bounces it, it rejects it back out. Right? So water comes in, it says, get out. Water comes in, it says, get out. Right? So it just keeps bouncing back and getting rejected. So water also needs to use a protein channel to come in. Right? Protein channels that move water have a special name, and that name is aquaporins. Aqua, meaning water. Right? Aqua, okay, water. And then porin, like the pores in your face, right? They're holes, pores are holes. So this is an aquaporin. Okay. And so we're still moving from high concentration to low concentration, but we need protein channels to help get it done. That's facilitated diffusion. Facilitated diffusion. Okay. And that's it for passive transport. Moving on to active transport. Active transport requires the use of energy. And most often when we are talking about active transport, we're talking about moving molecules from a low concentration to a high concentration, right? And that requires energy. And that's usually why we're doing active transport. There is another form of active transport that can move it either way, right? And I'll talk to that in a second. But first I'm going to talk about the typical style that we're talking about. Okay? So this is how we move a molecule from a low concentration to a high concentration. We use a pump. It's an active transport pump. And this is a protein channel. Okay? And it's designed specifically to push molecules across the membrane against the concentration gradient. So we are taking molecules from the area where there's a little bit of them, and we're putting them to the area where there's a lot of them, which is against the natural flow of things. So if we scroll way up to the top of my model, where I decided to put my active transport pump model, boom, okay. Here she is, right? So here's, again, the membrane, right? We're looking at the phospholipids and some protein channels. So this one that I've doodled here is my active transport pump. I've zoomed in on it over here to show how it works. Okay. So again, we're moving from an area of low concentration to an area of high concentration. So let's put a lot of triangles inside the cell down here. Right? So let's say there's a lot of triangles down here. Oh, and then I lost my internet connection. Just on the board though, it does that sometimes. Clearly I still have internet connection, but it's just for the whiteboard. Anyways, so pretend I got a lot of triangles down here and very few up here. Okay. So these channels have very specific shapes that only fit very specific molecules. And so this channel will fit triangles. So if the triangle plops in here, it connects and it's ready to go. But if a different shape, like a square, were to go in here, it wouldn't fit in. Okay? So only a triangle can go through this. So the triangle floats in and connects to this protein channel. Check mark. So this is what it's looking like. It's hanging out in this loading dock area. We then need to put energy into this situation. Right? We're forcing this molecule to move somewhere, so we need energy. That energy is going to come in the form of ATP. So I attach the ATP right to the protein channel. Right? They get attached. Okay? The P from the ATP is going to stick to the protein channel, and then the ADP that's left over is going to leave. Bye. Sticking this P onto the channel changes the shape of the channel. Right? So I went from being this configuration to now being this configuration. And so now it's open on the inside and closed on the outside. The triangle will now move forward and down, right? And now the triangle is down here. Eventually the P will fall off by P, and the protein channel will go back to its original shape. Right? When it goes back to the original shape, the triangle falls out and it goes to the other side of the membrane then, right? So it started up here, and now it's down here. And this is how it happened. Okay? So triangle engaged, ATP attached to protein channel changes shape, pushes triangle down, releases triangle into cell. Okay? Active transport. I can move molecule from an area of low concentration to an area of high concentration using an energy source, typically ATP. And last but not least is the other form of active transport, which are endo and exocytosis. And this is when our cell needs to move a very large molecule, too big for protein channels. So if it's exocytosis, it's moving that large molecule out of the cell. If it's endocytosis, it's moving that large molecule into the cell. And they both work by the cell membrane wrapping up the molecule and then plopping it to the opposite side. Okay? So the cell membrane will actually extend out around the molecule, surround it in a bubble of membrane, and then pass it to the other side. And so if we look at this model that I have here, okay, I got my cell, I'm trying to do some endocytosis. So I'm trying to get this large molecule into my cell. Okay. So the large molecule bumps into the membrane. Okay. The membrane invaginates a little bit, it dents in, and it makes a little pocket for my big molecule. Okay. It starts surrounding the molecule, so the membrane is now extending out and surrounding the molecule. That little invagination pinches shut, so now I'm completely surrounding the molecule in membrane. Okay. And then that membrane bubble pops off, and now I have a vesicle, which is a membrane bubble, holding that molecule, and it's inside my cell. Okay. So this takes a lot of energy to do for the cell, so this is using a fair amount of ATP, which is why it's active. But the movement against or for the concentration gradient here really doesn't matter. It could be going with the concentration or against it. It's just whether the cell wants to put this material inside or outside, and that's it. Right? And so if it's like, you know, a hormone is trying to get to go to a different place, it might be putting it out. If it's a nutrient it's trying to take into it, you know, it might be taking it in. Endo and exocytosis. So yeah, that's it, right? I got two forms of passive, which are simple diffusion and facilitated diffusion. Neither use ATP, right? They don't need energy at all. They're moving from a high concentration to a low concentration. And I have active, which is always using energy to move a molecule. Usually, it's to move it from a low concentration to a high concentration using the active transport pumps. But it can also be this process of endo and exocytosis, which we use to move very large molecules by surrounding them in membrane and then passing them from one side to the other. And that's it. So yeah, cool, awesome. Have a great day. Bye.